Hello, Bacho. Let us look into the question. The question is saying that we have to match the following enzymes with the functions in DNA replication with their respective function. So let us look into it. The key concept is DNA replication and enzyme related to it. So let us look into the DNA replication process and enzymes associated with it. Suppose this is a DNA which is uncoiled by the help of topoisomerase, and what happens is that over here helicase is found that help in the uncoiling of the DNA, and it helps in the releasing of the hydrogen bond present between the two nucleotides or nitrogenous bases. Now, since helicase, first of all, the first enzyme found is helicase, it helps in unwinding. The second enzyme responsible is polymerase. Since it is associated with DNA replication, it will be DNA polymerase. It helps in the addition of new nucleotide to the growing chain of DNA. Apart from that, when we talk about the clamp, it helps in stabilizing this polymerase over this DNA segment or strand. It helps in stabilizing the polymerase clamp. It stabilizes this. When we talk about the SSB, the fourth enzyme that is present in this process of DNA replication, what SSB helps in that is that it binds to the single stranded DNA. Suppose it is binding to the single stranded DNA, and it prevents the DNA from rebinding or reassociating the two strands with each other, so that the DNA replication can take place effectively and efficiently. So, SSB, which is single strand binding protein, it helps in the maintenance of uncoiled strand in the single form, the uncoiled single strand. So, these are the properties. Now, let us look into the columns. First column, DNA helicase. DNA helicase, it also known as helix destabilizing, because ये स्टेबलाइज नहीं कर रहा है बल्कि इसको डीस्टेबलाइज कर रहा है और इसको खोलने, it is uncoiling the DNA. That is why it is called as destabilizing enzyme, and it unwinds the DNA double helix at replication fork. This is a replication fork because it looks like a fork-like structure. Now moving further, polymerase, it helps in building a new duplex DNA strand by adding nucleotide in 5' to 3' direction. It also performs proofreading and error correction. So these are the functions of polymerase itself. So B will be belonging to 4. Moving further, DNA clamp. DNA clamp is basically a protein which prevents DNA polymerase third from us dissociating from the DNA parent strand. सो ये जो पॉलीमरेज जब यहाँ पे असिस्ट होता है तो ऑलवेज द clamp help the polymerase to stay at position and prevents the DNA polymerase third from dissociating from the DNA parent strand. Single stranded binding protein, we have talked about it, that it binds to single stranded DNA. So the answer that shows the coherence with these following columns is that helicase is for 3, A3. So this will be the option A3, B4 polymerase, B4, C1 clamp is for this one, and D is for 2. This is also very right. SSB is for bind to single stranded DNA. So this is the final explanation. Hope you all understood. All the best.